Yeah, everyone can see the slide?
I think so. Yep, we should all be able to see it.
Okay.
Thank you, everybody, for joining us for our honors program colloquium here in the spring of 2024. We are very excited to be welcoming my colleague, Dr. Sebastien Schmidl, who's an associate professor of biology and the coordinator of our RELLIS program down in Bryan, Texas, I think it is. So today, Dr. Schmidl is going to be speaking to us on Frankenscience, and we should have some time afterwards for questions. So I'm going to go ahead and mute myself. If everyone else would like to do the same, we will turn our attention to Dr. Schmidl.
Thank you so much for the introduction. I'm really happy to be here. That's always one of the few chances for me to talk to all of you in Texarkana. As Dr. Nakashian just mentioned, I'm now with A&M Texarkana for 6 years, and for the same amount of time, also the coordinator of our program here outside of Bryan College Station. For everyone who's maybe not too familiar where exactly it is, so basically, here's the College Station in Bryan, here's the A&M main campus, and the RELLIS campus is just here outside of the city. And it was basically an old airfield. So Buzz Aldrin was trained here before he actually went on the Apollo missions. And yeah, it was, I think, A&M owned this whole property for years. They didn't know really what to do with it. And yeah, 2018, they started to offering through the system schools bachelor degrees here. And yeah, we are from A&M Texarkana. We're offering biology here, and as well as electrical engineering, and then the other system schools offering other degrees in business, in criminal justice, and so on. And the nice thing is it's a really new campus, so everything is new. There's basically a building every half year that they're just finishing it. Here's the a quick overview. This is the building actually that I'm in right now. This one is a picture from the beginning of last year. Now we have already another building basically here. There's just the construction just finished. And then they start another one over here. And here will be a new STEM building ready in fall of 2025. So it's really one of the, I would say, fastest growing campuses in Texas right now. They spent over $1 billion just in infrastructure. And the idea is really to have more the collaboration between our programs and the industry. So we try our best to get in some industry partners here. And there are also some biotech companies. That's also why I focus today's talk more on the biotechnology side of my research, because that's also where there's more and more interest from the student side. And here you see also some ideas about the labs that we're having. Like I said, it's a new building. The building opened in 2020. And here you see some of the students who are doing research with me. And and I will continue a little bit more about my research interests, which you see basically here. There are basically two main themes. The first one is one which was building up on my research, which I've done in my undergrad and grad studies, which is mycoplasma research. Specifically, I'm interested about the adaptation and the thriving of microorganisms at the edge of life. And mycoplasma species are really nice to use for this kind of purposes. They have small genomes, but nevertheless, they're still able to do independent life. In addition to this one, also they can cause infections in most of the host organisms. And that's why they're really great little bugs to study when it comes to see, okay, what do you really require? What are the requirements for life in general? But today I want to focus a bit more on my second basically major interest, which is how can you design genetically engineered bacteria? And specifically, I'm interested in signaling pathways and how you can engineer signaling pathways in a way that they can help you to sense specific environmental pollutants, for example, or other biomarkers that you can find maybe in the human microbiome. Okay, but before we start a little bit with this, more like the bigger scope. So when we're talking about engineered bacteria, there's basically two main themes where you can use them for. So one was basically using it for chemical factories. And a lot of these bacteria basically are really great when it comes to production of drugs, fuels, food additives, but even chemical feedstocks. But even beyond this basically chemical factory research, there are also other specific tasks that you can really tackle with the use of engineered bioorganisms. And this would be, for example, bioremediation, which is a huge field. For example, when it comes to oil spills, heavy metal pollutants, any other toxic pollutants in the soil environment. And a lot of research was really focusing over the last 10 to 15 years to figure out how can you engineer organisms to basically get rid of these pollutants? And then the other booming field really in recent years is the therapeutics and diagnostic field. This also is where most of my research is really directed towards too. And in addition to this one, also then the basic science field. Okay, so how can you use engineered organisms to study genetic circuits? How is gene regulation really working in microorganisms? And how do they really adapt to specific environmental conditions? And specifically, I'm interested in two component systems, okay? Which I always say it's basically the eyes of microorganisms. So they don't have real eyes, they don't have a nose, they can't smell anything around them, but they need still a way to figure out, okay, what's going on in the environment. And one way to do it is by using these two component systems, which, like the name is always saying, they're made of two components. So we have always here a sensor kinase here, and then we have also so-called response regulator. In each of these two proteins itself is also then split into two different domains. So the sensor kinase has a sensor domain itself, which is focusing towards the outside of the cell. And this one is basically important for sending any kind of input signal. And then we have also the cytosolic kinase phosphatase domain, which is really important because this one is doing an autophosphorylation upon sensing any kind of stimulus. And then this phosphate, which is attached to a histidine, is getting transferred to the response regulator, which typically upon phosphorylation causing a dimerization, and this dimer can then bind to a DNA stretch, which is called the promoter. And the promoter is really important because without the promoter, no gene could get expressed. Okay. When it comes to gene expression, really, these promoters are essential. And response regulators, in this case, they're binding to promoter regions and helping then the RNA polymerase to start the transcription process on the long run. And as you see here, the response regulator itself also has the receiver domain in order to basically get the phosphate attached to, and then the actual DNA binding domain, which is really important for the interaction with the DNA itself. Okay, and when you look at these systems, they are not relatively unique to only certain bacteria. You can find them all over the bacteria kingdom, really. And each organism has either a couple of ones up to really 200. And they can sense a couple of different molecules. It can be chemicals, but even can be physical inputs when it comes to light, which we'll talk a bit more about in the next slides. But there's really a huge variety of input signals. And then in the end, most of the time, what you see is a change in the expression of certain genes, which typically helps the bacteria to adapt to new ecological niches, or just basically maybe fighting off even some other rivalry bacteria, which are living maybe in close proximity. The systems that I'm really interested in are the ones that you see here on the next slide. And these are the ones which are really important when it comes to sensing light, which is really unique, if you want to say it like this. And most of these systems can only be found in cyanobacteria. So here, for example, shown Synechocystis, which is also commonly known as the freshwater pond scum. You can probably isolate out of the pond outside of the main campus. I'm pretty sure you can find it there too. And they use these two component systems, a specific version of it, which has also these chromophores here attached to it, which helps them at the, which serves as a light harvesting complex. And this system allows them to change the gene expression in a way that they can really adapt their photosynthesis to the specific wavelengths of light that they're seeing. So for example, when they're more at the higher depths in the lakes, they need different systems in order to have a maximum photosynthesis. But when they're by the lower depths in the lakes, they need different other systems. And they use these two component systems in a way that they can really always have maximum photosynthesis, ATP production, and they can grow best. Okay. And when it comes to the system, they have a couple of ones you see here, which can basically sense almost all the different wavelengths of light in the visible spectrum, but also a couple of ones in the near infrared spectrum as well. And these systems really are really great tools when it comes to engineering, but also for the industry, because they have a couple of advantages over these more traditional systems, which maybe can sense chemical inputs. And this is basically, it's a non-invasive control mechanism. You can shine light on the microorganism. It's not doing anything to them. So they're not actually, it's not causing any deaths of them. They're not getting lysed and stuff like this. It also allows you a spatial temporal position control, which means you can shine the wavelengths of light. You can turn off the light source and they're stopping gene expression, turn it on again. They start with the gene expression again. Then there's also no or any minor side reactions when it comes to this, because they're not relying on any chemical inputs that you have to expose the cells to. So light is really not toxic to the cells. There's no side effects that can really happening. And the other thing is also it's really stable in vivo. I mean, you shine wavelengths of light and actually a constant wavelength, not using these wavelengths. If you use some of the other chemicals, they can utilize them. So there's always a fluctuation when it comes to the concentration itself. And because of this, really, these systems attract a lot of interest, not only from the research side, also from the industrial side. Okay, because there's always a need of microorganisms which can produce certain compounds, like we talked before, like drugs, food additives, in really high quantities. And traditionally, these industry would have used chemical inducers. So they would have chemicals to it, which was basically giving you side effects. They were metabolized. You don't have constant expression. And the other thing also, it was really expensive. As you can imagine, you have to buy all the chemicals. You have to always make sure to add the chemicals in the right amount to make sure everything is constant. With light, you just buy these LEDs really, and then you can use them really for long periods of time. There's really a huge saving when it comes to the cost of the production of systems. And the system that I'm specifically interested in are these light systems. And there's two ways how you can really engineer them in a way. The first approach would be you use a native signaling pathway in the organism where you would like to study them, or would you like to express a certain protein. So typically we use E. coli, which is the model organism for all these kind of expressions. You use one of these native pathways and then build one of these hybrid sensor kinases in a way that you basically fuse the sensor domain of one that you would like to study to the kinase phosphatase domain of the sensor kinase. And then basically upon receiving the stimulus, you get an output signal in this way. This one was long used. It was really successful. However, it has some disadvantage as well. As you can imagine, if you use a native system here, it always interferes with the native signaling pathways. So if this two-component system is, for example, linked to the stress response, if you turn on the signal, the cells think they're in stress. And this always, of course, causes problems when it comes to growth. And that's why this one is not really the ideal approach, because you mess really with the internal metabolism of the bacteria itself. And that's sometimes not really what you would like to have to happen. In addition to this first approach, the second one would also be you just transfer a system that you found, for example, in a cyanobacterium, and completely transfer it into E. coli. Okay, this one shouldn't have any of these negative effects here. You're not messing with the internal metabolism. You just bring in a completely new system, so everything is new. However, one issue in this one is always the expression of these systems. So if you just copy and paste a piece of DNA from a cyanobacteria, put it into E. coli, you cannot really be sure that it's expressed, okay? Because there's some optimization necessary when it comes to codon usage. Not all the codons are used equally in all the organisms. And the other thing is also this promoter here that you bring in, which is important for the expression of specific genes, might be silent. Okay, so it maybe interacts or maybe it's recognized by the RNA polymerase in the specific host organism. But if you bring into another organism, this new RNA polymerase doesn't recognize the specific motifs which are conserved in these promoter regions. So that's why this approach seems to be really the way to go. However, there's also some tweaking really necessary when it comes to studying these kind of systems. The system that I used a lot are the two shown here, which actually are built on both of these approaches. So the first one, what you see is a so-called far-red transcription device, which is based on these hybrid sensor kinase, which you see basically here. And they use the original ENVC OMP-R system, which you can find in E. coli. And then they built by this new hybrid kinase, which is called CIF8, which has a sensor domain from a cyanobacterium, from Synechocystis, and the link to this native pathway here, which is important for the stress response. Okay, and by doing this, basically, you can turn on the system by shining far red light on the bacteria, and you can turn off the system by shining red light to them. Okay, so it's a great, basically, transcription device, gives you output by just shining far red light, and it's basically based on the first approach. Then for the second approach here, we have the CCAR system, and this one was basically wholesale transfer. So both of these proteins, CCS and CCR, are originally from Synechocystis. You brought them in, and then you have a device which is basically getting turned on if you shine green light, and it's getting turned off if you shine red light to them. And again, then you can measure the gene expression in response to this different kind of wavelengths of light. And these systems were already built relatively early in the early 2000s, really, and they were widely used for these kind of funny, really, experiments that you can see here, which is bacterial photography. So you bring them into E. coli, and then you shine specific wavelengths of light into E. coli. E. coli then expresses here a specific enzyme, which is able to degrade a black pigment in the plate. And then you get these nice contrast images on a plate. And that's also something what I like to do with some students in the lab. And it's really easy to do really quickly. And they can see basically how you can influence gene expression relatively fast just by switching different kind of wavelengths of light. And these kind of experiments work really for both. And yeah, for these kind of gimmick experiments, the system works really well. But there were some other really major issues, which you see here on the next slide. Okay. And I would like to focus first here on this graph. So also the system worked. You see here the switch. So we're talking about CCSS and CCAR. So in red light, there's still some leaky expression, but at least you get a 6-fold induction if you change from red to basically green light. But that's not ideal for the industry. Okay, for the industry, what you want to have is specifically if you express toxic compounds, you want to have here really no leaky expression. So the system should be here completely off and should here relatively maximize really the expression level. Okay, so you don't want to really work in some gray zones. You want to have more like a black and white situation where the system is completely off at one and then completely on the other hand. Okay, and that's what's really where I was brought into this project here in order to figuring out, okay, how can we really engineer these systems to make them more useful really for the industry? And yeah, what you see here are these the original plasmid designs. So for everyone who's maybe not a biologist, mine looks like maybe really cryptic, I would say. But what you basically hear is some extra chromosomal pieces of DNA, okay, which can exist in E. coli in addition to the host chromosome. And this is really nice because these allow you to engineer or add specific genes to these plasmids, and then you can transform them into E. coli and you can express in your specific genes. This really gives you the benefit that you don't have to mess around with the chromosome itself. You can just basically engineer everything on the plasmid side, which are also smaller, and you can add specific genes to them. So for today, you can basically skip this one, which is important for the phytochrome production, which is important for light harvesting. But we're just focusing basically on this plasmid here, which basically just harbors the whole chunk of DNA from Synechocystis, which has a 2-component system. So what they did here is they cloned this chunk of DNA into the plasmid, which you see basically from here up to here. The only modification was done is in Synechocystis originally, the gene which is expressed is CPCG2. And yeah, the gene was actually here replaced by GFP in order to have a reporter output. That's also what you see here. You see here the expression levels of GFP in E. coli, because there's not really a good enzymatic assay for this protein over here. So it's useful in Synechocystis, but doesn't have really any specific function. We're talking about it in E. coli itself. So we just replace this by GFP. Then we have an output or a gene that we can read as an output signal. And this one gives you a nice green fluorescent upon the expression. And it's really widely used. Originally, it comes from jellyfish. It's important for the mating of jellyfish and stuff like this. And the Vibrio species you can find there. But it's really widely used as a reporter gene and research for tracing the expression of certain genes, but also for localization studies, if you're interested to know where certain proteins are localized inside of a cell. And yeah, this was basically the only thing what was done here. And basically, I just replaced CPCG2 by GFP, and you see basically you get this somehow leaky expression, which is not really ideal. Okay. To do this kind of experiment also, you see, you need actually a special type of equipment. And I don't ask me too many questions about this one. I'm really thankful that I worked really with some great engineers together who actually built the machine. And it's called the Light Tube Array, or just LTA, which is basically a shaker incubator where you have LEDs at the bottom and you have different wavelengths that you can shine then at the cultures while you're shaking your tubes. The tubes also have this foam around them, so they're completely in the dark if you're not shining any wavelengths of light at them. And this one allows you to grow your bacteria under specific light conditions. Okay, and that's really, it's a great tool because otherwise, if you just would grow them under ambient light, they would get all different kinds and you couldn't do any kind of these experiments. So you need really to grow them in the dark and only let them grow under certain light conditions, which you can adjust with the help of these LEDs here at the bottom. Okay, and by using this device, we will actually be able to really engineer these plasmids in a way that we were changing the expression levels of the kinase, which you see basically here, and also the response regulator on the other end. Okay, and by changing the expression level, you could then create this kind of heat plot where you increase the expression level of the response regulator, then you increase the expression level of sensor kinase, and you try to figure out, okay, where is there some kind of a sweet spot where you get not a lot of leaky expression, but you get actually the system completely turned on. So basically, where's the fold induction really maximized in this case? You build basically a whole library, different promoter regions, different ribosomal binding sites, which also affecting the expression. And then you look, okay, where is the sweet spot for the system that really it's useful for the industry at the end of the day? Okay. And yeah, this one is what you see here. So we re-engineered output promoters, optimized expression of the kinase, of the response regulator, optimized also even expression of the chromophore itself. And then you see the original system here from the 6-fold, where you were able to 117-fold. There's barely any leaky expression seen anymore now, and we could even increase a little bit the expression when the system is turned on. In a similar approach, we also use basically here for the red far red system, where you went from a 9-fold increase in expression now to a 70-fold expression. Okay, and this one is really now useful for the industry. You can express proteins, you can express toxic proteins even in really small amounts because there's no leaky expression in the system itself. And after we have done with this one, we thought, okay, now we have optimized the system. So why not go one step ahead and see, okay, where are some kind of limitations when it comes to engineering these kind of systems? Like I mentioned before, most of the research we focused on the sensor kinase side, right? How can you maybe do hybrid kinases, which are then basically feeding into native systems? But one issue is these kinases are relatively large, and they're really problematic when it comes to figuring out, okay, how do you find the right split point? And that's why we came up, okay, why not do rewiring on the response regulator side, which you see basically here. They're either way more modular. As you see here, most of them have a DNA binding domain. You see basically the winged helix, HTH, and the FIS domains. They're all basically DNA binding domains. But there are even some other domains which you can attach to these response regulators. Like there are a lot of here metabolic enzyme activities you can attach to them. So they're not only transcription regulators per se, they're also metabolic enzymes. They can even phosphorylate other ones, dephosphorate other enzymes. And because there's already this modularity in it, so why not try to re-engineer these systems on the response regulator side, which gives them some new ideas or new ways how we can study these systems. One way would, for example, be, so you have a new sensor kinase and you don't really know what is the input signal, but you would be really interested to know, okay, what are the input signals? What are the sensing? How does the regulation work? So you basically then transfer this kinase into E. coli, use your engineered output system, which we used before for the green and red system, this basically the CCR, CCS system. And then you can basically characterize a whole array of different sensor kinases and see what kind of inputs they're sensing. Or you can go the other way around. If you, for example, transfer the signal to another organism, you can now use your light input signals and can drive the gene expression and can see which genes actually, in this case, regulate by certain response regulators, okay, which is basically completely different to one before. Here, sensor kinase characterization. Here, you characterize more the gene expression side. And we used our light system again here as a proof of principle. So we had the optimized one. And then we built a whole library basically here of hybrid response regulators. So in total, we actually created 30 split points along the alignment between these two response regulators. So you see the alignment between OMP-R and CCR, and you see the different amino acids in their sequence. And then basically send wherever you think, okay, this might be a good point to make to create this hybrid. So basically you then created this new kind of proteins, and you see how is the light response then to far red light and red light, even though the system is originally basically from the green red system that we used before. And you see basically for some split points, specifically the ones around the linker regions, you were able to create functional hybrids, which are now responding to far red light and red light. And this was just shown as a proof of principle. Really, you can re-engineer not only the sensor kinases, you can also re-engineer in this case also the response regulators. And nobody wants to restore these split points every time. So for the fauna experiment based on this approach, then we just picked the best ones, right? The ones right around these linker regions, which seem to work pretty well, and want to see, okay, we know now everything works for our light responsive systems, but how is this whole approach looks like when they're talking about other two component systems? And one other one, which was relatively good studied, there's lots of research in E. coli, is one, the so-called NAL system in E. coli, which is important for sending nitrate. Okay, here again, you see the alignment between the different response regulators, the specific split points for that we tested here in this case. And then as the output, we use different other promoters that were also known to work really well in E. coli. So we use the response regulator or the DNA binding domain of LIR and UHPA together with the specific output promoters. And as you can see basically here, that now these systems are able to respond to nitrate when you actually have a certain, when you actually look at this certain basically here hybrid that you created. Okay, so again, similar to experiment before, the system or these hybrids only work when it comes to light sensitive systems. Even when you use a chemical input system or use a chemical two component system like the NAL system here in this case, the nitrate sensing still works. And this hybrid response regulator even drives then the output of specific promoters based on this really Frankenstein or hybrid really response regulator in this case. Okay, so all these experiments so far were done in E. coli. Okay, and you can imagine now the next step would be, okay, we know we can do this with physical stimuli, with chemical stimuli in E. coli, but how is this whole system working when it comes to other organisms, right? Not everyone likes to study E. coli, not everyone is interested in E. coli. So how is it working, for example, use Bacillus subtilis, which is the other great model organism in the bacterial world. So most experiments are done either in E. coli or Bacillus. So in this case here, we first basically transferred the whole system from E. coli into Bacillus subtilis. And as you can see here, nothing is working. Okay, no response, no switch basically. If you add nitrate, no nitrate, no response to this one. However, if you build a hybrid system, which is using then the DNA binding domain together with the promoter, which are native to Bacillus subtilis, then the system may be able to sense nitrate in a really specific response, which is here in this case. And this one showed us really that our approach not only works in E. coli, it also works actually in other organisms, and nitrate sending is not possible here with the help just with some little tweaks by using basically DNA binding domains and promoter regions, which are more native to the organism itself. Okay. And yeah, one of the next approaches then was basically trying a big scale experiment. So Shevanella audiensis is another really important microorganism in a way that it's often used for getting rid of heavy metal pollutants in the soil. Okay, so it's an important organism for bioremediation. And what is with this organism that has lots of two component systems, but there's not much known about, okay, what are these two component systems sensing? And in order to characterize them, we built a whole library. So basically, we cloned all these sensor kinases into E. coli, and then also we built basically specific hybrids of the response regulators, and then basically screened all these systems here for, as you see, a whole panel of different metabolites, which we thought would make sense to the environment where typical Shevanella is growing, which is like a soil aquatic organism. And by seeing this one here, there might be not the best fold inductions, but you see here, at least you can narrow down that some of these chemicals here or stimuli actually inducing a specific signal, which then gives you hints, okay, this system could be important for sending this kind of stimuli. And then actually you can do further studies in order to narrow it maybe more down or see, okay, how the regulation works in general. Okay, really important here is we didn't build basically specific libraries again when it comes to split points. So we just picked here the best one from our experiments before. And it's a little bit a gamble, as you know, probably because we were lucky that actually it worked in this case. Okay, but it just shows you really the potential here. You can basically screen organisms, the two component systems. And in the case of Shevanella, this organ is not really easy to grow in the lab. So doing this kind of studies in Shevanella directly is not possible, also because there are not genetic tools available. But doing it in E. coli is a great way really to study this organism further. And one of the next really research approaches would be then going into the human microbiome, which is in the news everywhere. Everyone knows about the gut microbiota. If you go to the grocery store, the pharmacy, you see all the probiotic pills that they're selling, to tailor your gut microbiome, to have more healthier lifestyle. And this is really important in recent years because it is more and more clear that the gut microbiome, the composition of microorganisms in your gut, they're really important only for type 2 diabetes and obesity. They're also associated maybe with diseases which you would not really think that microbiota in your gut have a role with, like anxiety, depression, and even certain types of cancer. The problem with all these gut microbiota is there's not much known about them. A lot of them you can't even grow in the lab. They have so specific growth conditions that you cannot really use them as a model organism for lab experiments. And you need really another host, like E. coli, for example, to get a little bit better idea, okay, how are these organisms sensing the environment? What kind of regulatory mechanisms are going on in there? And then basically try to build some, you see later on, some smart probiotics, okay? And here's basically the framework that we're currently working on. And we're looking in literature and looking, okay, what kind of diseases are out there? What data about DNA RNA expression are there? What metabolites are associated with certain diseases? And then we're trying to engineer, okay, what kind of pathway, similar to the examples before, can be built by mining specific parts of these organisms? Okay, and then the idea is really to study really how they're sensing, how gene transduction is going on, how they're responding, what are they producing, and building the system in the lab in E. coli specifically, doing specific tests to see, okay, what the sensor inputs, similar to the experiment with Chavanella, and then learn really a little bit more about the microorganism itself, which are in which close proximity are they with other ones, what are their needs when it comes to growth, and so on, which helps at the end of the day basically to engineer new therapeutics and diagnostics. And some examples, which is basically a long shot, is really shown here. When it comes to the therapeutic side, is okay, how can you build an organism which can sense disease biomarkers maybe in your gut, and then immediately releases a therapeutic compound? Okay, you release it right at the specific location, which is way better as any kind of treatment that's available these days, because you're basically taking them orally, but then they're basically maybe at the specific location where they needed, they are so much diminished that they barely make any effect. Okay, but having an organism which only localized expresses certain compound is really the way to go, and maybe even tackle certain diseases in this case. And on the other side, diagnostically, the other thing is also, how can you engineer maybe a probiotic pill which you just ingest with your food, measure a certain compound in your gut, and then basically you take the stool sample and see, okay, get a positive or negative signal for certain diseases. And this can give you basically like a warning already, okay, maybe you should go to a doctor, there's something maybe wrong with your gut microbiome. And early prevention really helps in the development of these kind of diseases. And for both of these, really, getting the right parts is really the key challenge. Okay, we need two component systems to sense, okay, disease biomarkers, disease compounds produced from other microorganisms. And that's why this whole framework, what I showed you today, is so great, because you can do all these characterizations in E. coli. You don't have to go into the native organisms, which are difficult to grow. We have no understanding how they're growing. You can do everything in E. coli, where you know all the genetic tools, and yeah, everything is really available in this case. And yeah, this was already everything I've planned today. Just a shout out here to my collaboration partners, and of course also all the students who help working with me in the lab, not only on this project, also on the Mycoplasma project. And I'm here to answer any question that you might have.
Okay, wonderful. Thank you, Dr. Schmidl. So we'll open it up to questions. I mean, if you've got one, feel free to unmute and just go ahead and ask. Or you can drop them in the chat if you want me to read them for you. Not all at once, just one at a time. One at a time.
I actually have two.
You may ask them both. That's right.
If that's okay.
Yeah, of course.
He knew I probably was going to.
I was counting on it.
You were talking about this bacteria actually removing the heavy metals from the soil?
Well, when it comes to heavy metal pollutants, it's still basically they're building small models inside of them. The issue is still with all these microorganisms used for heavy metal or getting rid of heavy metal in the soil is how are you getting rid of the bacteria itself, because they're forming typically the inclusion bodies inside. And then, yeah, after a while, when the cells are lysing, they're releasing the heavy metal itself. So in literature, often when they're talking about bioremediation, it sounds like this fairy tale story. They're just going there, getting rid of all the heavy metals. What they're never telling you really is they're typically then stored inside of the bacteria. So you have to figure out a way how to get rid of the bacteria without releasing them again. And that's why I always say these organisms that I showed here, also like Shevanella specifically, they can deal with heavy metals. They're then producing inclusion bodies. So the soil is not exposed to it anymore, but they're still there in the environment, technically.
I was thinking that very thing when you were talking about that, because that's they've tried that with the plants, and of course the plants can remove the pollutants, and then they feed it to cattle, but they're not really getting rid of it. They're just moving it from one place to another.
Yep, that's always the thing.
That's I've seen some work that they're actually doing in Israel right now with the E. coli as far as removing gases like CO2, and then also working on with heavy metal, but they're having problems because what's coming out the other end of the bacteria.
Yep, that's always the thing.
What they're eating, so
you just store it somewhere else.
Yes, exactly. Yep. And then the other one, because this is very interesting, the gut microbiome. You said that you were unable. It has very specific growth requirements.
Yes.
Therefore, it's hard to grow in the lab. What about what about sequencing that? Would that?
That's why most of the research these days, when it comes to gut microbiome, is on the metagenomic side. You basically just take samples from specific patients or other individuals, and then sequence basically all the microorganisms that are in there. And that's how we know so much about the gut microbiome. So the sequences are available. You can use them with our framework, but the organism itself, you cannot really grow them. And even when it comes to probiotics, what is always funny, so these companies selling you probiotics, and they know that certain organisms are beneficial for us, but they don't know why they're beneficial. So there are not really any conclusive studies because they cannot grow them. They just know, okay, if you get this, or when you have this microorganism in your gut, it tailors, or it basically keeps away the bad ones. And that's basically the good thing. But how do they sensing it? How are the growth requirements? So everything what we know is only from the metagenomic side, like similar to your experiment you're just doing right now.
Yes.
Okay. No, you're fine, man. Any other questions for Dr. Schmidl? Oh, okay. Dr. Torben asks, what is the primary application of your research in real world kind of scenarios, treatment, that sort of thing?
Okay. So when it comes to the light systems, so we had an industry partner who was we wanted to basically produce toxic compounds, and you need basically this on-off switch. Okay, so in the industry, one issue was really using chemicals is not beneficial because it should cost expensive. And if you do it in smaller scale, these light systems actually doing the trick, you can use them. And when it comes to the other mining things here is really just understand the basic biology of the gut microbiome. How do they sensing? What are disease biomarkers associated with certain diseases? Engineer the microorganism and then basically produce certain compounds which can maybe degrade these biomarkers, or basically give you an early warning detection system so you can go to the doctor and have an idea, okay, yes, there's something wrong with me, fix me maybe.
Okay, thank you. Any other questions? In that case, I will say thank you once again to Dr. Schmidl. It was very interesting. Join us in a couple of weeks for our currently final honors colloquium of the spring. You never know if anyone's going to jump in at the last minute. But Dr. Greg Hogan in chemistry will be presenting at 11 a.m. on April 23rd. So I'll send out some emails about that. But again, thank you to Dr. Schmidl. Thank you to everyone who has turned up. And I will go ahead and close it there for us. So thank you once again.